only two options: make progress or make excuses. If you want to uplift your life, you have to progress well. Or if you want to make excuses, just quit. Happy morning, dear students. I'm welcoming you all to this online class. In previous session, we started the 5th chapter, molecular genetics, and we completed up to restriction of DNA. Today, we will start with transcription.
Francis Crick proposed the central dogma in molecular biology, which states that genetic information flows from DNA to RNA and then to proteins. The DNA on transcription converted into RNA and by translation is converted into proteins. So what is transcription? The process of copying the genetic information from one strand of DNA into RNA. So transcription means copying DNA into RNA. This termed as transcription. This process takes place in presence of DNA-dependent RNA polymerase. In some retroviruses like HIV, the flow of information is reversed. RNA synthesizes DNA by reverse transcription, then transcribed into mRNA by transcription and then into proteins by translation. For a cell to operate, its genes must be expressed. The RNA that carries the genetic information encoding a protein from genes into a cell is known as messenger RNA. For a gene to be transcribed, the DNA, which is a double helix, must be pulled apart temporarily, and RNA is synthesized by RNA polymerase. This enzyme binds to DNA at the start of a gene and opens the double helix. Finally, RNA molecule is synthesized. The nucleotide sequence in the RNA is complementary to the DNA template strand from which it is synthesized. Both the strands of DNA are not copied during transcription. How? Why? We have to ask such questions. Both the strands of DNA are not copied, but only one strand are copied. Why? The reasons are, if the both strands act as template, they would code for RNA with different sequences. This in turn would code for proteins with different amino acid sequence. This would result in one segment of DNA coding with two different proteins. Hence, complication occurs in the genetic information transfer machinery. The other reason is, if two RNA molecules were produced simultaneously, double-stranded RNA complementary to each other would be formed. This would prevent RNA from being translated into proteins. So, only one strand of RNA is copied to DNA is copied to RNA. Next we see what is transcription unit and the genes involved. A transcriptional unit in DNA is defined by three regions: a promoter, the structural gene, and a terminator. The promoter is located towards the 5' end. It is a DNA sequence that provides binding site for RNA polymerase. In this 5' end only, the RNA polymerase gets binded to synthesize the RNA. The presence of promoter in transcription unit defines the template and coding strands. The terminator, that means the end region, located towards the 3' end of the coding strand, contains a DNA sequence that causes the RNA polymerase to stop transcribing. In eukaryotes, the promoter has the AT-rich region called Tata box. And in prokaryotes, this region is called Pribnow box. Besides promoter, eukaryotes also require an enhancer. The two strands of DNA in the structural gene of transcription unit have opposite polarity. DNA-dependent RNA polymerase catalyzes the polymerization in only one direction. The strand that has the polarity 3' to 5' that act as a template and is called as template strand. The other strand which has the polarity 5' to 3' has a sequence same as RNA except thymine instead of uracil, because DNA contains thymine and RNA contains uracil and is displaced during transcription, and this strand is called as coding strand. So what is template strand and coding strand? Template strand means 3' to 5' act as template, that is called template strand. The other strand, that is 5' to 3' that act as a coding strand. The structural gene may be monocistronic or polycistronic. In eukaryotes, each mRNA carries only a single gene and encodes information for only a single protein and is called as monocistronic mRNA. In prokaryotes, cluster of related genes known as operon. The cluster of gene is called as operon, often found next to each other on the chromosome or transcript together to give a single mRNA. Hence, they are called as polycistronic. Polycistronic is found in prokaryote and monocistronic is found in eukaryote. Before starting the transcription, the RNA polymerase binds to the promoter, a recognition sequence in front of the gene. Bacterial RNA polymerase consists of two major components, the core enzyme and the sigma subunit. So, this bacterial RNA polymerase consists of two components. One is the core enzyme, another one is the sigma subunit. The core enzyme beta 1, beta and alpha is responsible for RNA synthesis, whereas a sigma subunit is responsible for recognition of the promoter, which promoter is binds at the promoter site. Promoter sequence vary in different organisms. RNA polymerase opens up the DNA to form the transcription bubble. The core enzyme moves ahead, manufacturing the RNA, leaving the sigma subunit behind at the promoter region. The end of the gene is marked by the terminator sequence that forms a hairpin structure in the RNA. The subclass of terminators require recognition protein known as the Rho, that means like this symbol, to function. Next, we see process of transcription. In prokaryotes, there are three major types of RNAs: mRNA, tRNA, and rRNA. All three RNAs are needed to synthesize a protein in a cell. The mRNA provides the template, tRNA brings amino acids and reads the genetic code, and rRNA play structural and catalytic role during translation. There is a single DNA-dependent RNA polymerase that catalyzes transcription of all types of RNA. It binds to the promoter and initiates transcription. So that is called as initiation. The polymerase's binding sites are called promoters. The polymerase where it binds in the DNA is called as promoter. It uses the nucleoside triphosphate as substrate and polymerizes in a template dependent fashion following the rule of complementary. After the initiation of transcription, the polymerase continues to elongate the RNA, adding one nucleotide after another to the growing RNA chain. Only a short stretch of RNA remains bound to the enzyme. When the polymerase reaches a terminator at the end of the gene, the nascent RNA falls off. So, also the RNA polymerase. The question is, how the polymerases are able to catalyze the three steps, that is initiation, elongation and termination. The RNA polymerase is capable of catalyzing the process of elongation. The RNA polymerase associates transiently with the initiation factor sigma and termination factor rho. Initiation factor is the sigma factor and the termination factor is the rho. To initiate or terminate, to start or to end the transcription respectively. Association of RNA with these factors instructs the RNA polymerase either to initiate or to terminate the process of transcription that you can see in this picture. In bacteria, since the mRNA does not require any processing to become active, and also since transcription and translation takes place simultaneously in the same compartment. Why transcription and translation occurs in the same compartment simultaneously? Since there is no separation of cytosol, that means cytoplasm and nucleus in bacteria. That means there is no nuclear membrane. Many times the translation can begin much more before the mRNA is fully transcribed. This is because the genetic material is not separated from other cell organelles by nuclear membrane. So what is the reason why bacteria undergoes transcription and translation simultaneously? Because the genetic material is not separated from the other cell organelles by a nuclear membrane. In eukaryotes, there are at least 3 RNA polymerases in the nucleus. That means in addition to RNA polymerase found in the organelles. So there is a clear division of labor. All will do different works. The RNA polymerase 1 transcribes rRNAs, whereas RNA polymerase 3 is responsible for transcription of tRNA. And the RNA polymerase 2 transcribes the precursor of mRNA and the heterogeneous nuclear RNA. So these are the functions of RNA polymerase 1, 2 and 3. In eukaryotes, the monocystronic structural genes have interrupted, that means interrupted coding sequence that is known as exons. The monocystronic structural genes having interrupted coding sequence is called as exons, expressed sequence, and non-coding sequences are called as introns. Coding sequences are called as exons and non-coding sequences are called as introns. The introns are removed by the process called splicing. What is meant by splicing? So the introns are removed by the process called splicing. hnRNA, that means heterogeneous RNA, nuclear RNA, undergoes additional processing called capping and tailing. Capping and tailing. In capping, an unusual nucleotide methylguanosine triphosphate is added to the 5' end, whereas the adenylate residues, that means 200 to 300 poly A, are added to the 3' end in tailing. In capping, unusual nucleotide is added in 5', whereas the adenylate residues are added in 3'. Thereafter, this processed hnRNA, now called mRNA, is transported out of the nucleus for translation. The split gene feature of eukaryotic genes is almost entirely absent in prokaryotes. Originally, each exon may have coded for a single polypeptide chain with a specific function. Each exon, that means the coding sequences, so is with a specific function. Each exon have specific function with single polypeptide chain, that means a single protein. Since exon arrangement and introns removal are flexible, the exon coding for the polypeptide subunit act as domain, combining in various ways to form new genes. If introns arose late, how did they enter into the eukaryotic gene? Introns are mobile DNA sequences that can splice themselves out of as well as into specific target sites, acting like a mobile transposon like elements. That means that mediate transfer of the genes between the organisms, horizontal gene transfer. This 8GT horizontal gene transfer occurs between the lineages of prokaryotic cells or from the prokaryotic to eukaryotic cells or between the eukaryotic cells. Next we see what is genetic code. The information needed to direct the synthesis of protein is contained in the mRNA in the form of genetic code. The genetic code is the system of nucleotide sequence of mRNA that determines the sequence of amino acids in protein. Codons are a group of 3 adjacent bases that specify the amino acid of protein. Crackling of genetic code was done by Marshall Nierberg and Hargovind Kurma. And this is the table showing the dictionary of amino acids, code words which is present in mRNA and other triplet codons. And there are around 64 codons, including these 3 stop codons. It is also called as nonsense codons. This codon which play least role in the amino acid than the first 2 beats. They can read from 5' to 3'. If you see the characteristics of a genetic code, the genetic code characteristics is very important. That means the salient features of genetic code. The genetic codon is a triplet code. Each amino acid is represented by triplet sequence. There are 64 possible codon sequences. 3 of the 64 possible triplet do not code for any amino acid, so they are called as nonsense codon. Example for this is UAA, UAG and UGA. These are nonsense codons. And the other characteristics of genetic code is the code is almost universal. Exception to the universality of the genetic code are found in human mitochondria. UGA codes for tryptophan instead of stop codon. AUA codes for methionine instead of isoleucine. CUA codes for threonine instead of leucine. These are present in human mitochondria. The initiator codons. The example for initiator codons in eukaryotic and prokaryotes are: in eukaryotes, AUG codes for methionine, and in prokaryotes, AUG codes for N-formyl methionine. The terminator codons. Example for terminator codons are UAG amber, UGA opal, and UAA ochre. So these are some of the initiator and terminator codons. There are some exceptions related to stop codons. They are UAG, pyrrolysine, UGA, tryptophan that is found in the mitochondria of human, UGA that is selenocytosine which is the 21st amino acid found in the mitochondria of a human. So these are some exceptions. The another important characteristics of genetic code is non-overlapping and without punctuation. During translation, code is read sequentially without any spaces, taken 3 at a time. Example, if you read this code AUG, CUA, GAC and UU. So the codes are non-overlapping and without punctuations. And the other important characteristic of genetic code is it is degenerative but unambiguous. As there are 61 codons for 20 amino acids, one amino acid has more than one codon and the code is referred to as degenerative. Although an amino acid may have more than one codon, each codon specifies only one amino acid. Thus, the genetic code is unambiguous. So, if an codon specifies only one amino acid, the genetic code is unambiguous and it is a regenerative. The codons that designate the same amino acids are called as synonyms. Two amino acids, methionine, AUG, and tryptophan, UGC, each have only one codon. So, for synonyms, the codons designate with same amino acid. The remaining amino acids have multiple codons. Example, arginine. So, the codon anticodon recognition. The codons of the mRNA is recognized by the anticodon of tRNA. They pair with each other in antiparallel direction, that is 5' to 3' of mRNA with 3' to 5' of tRNA. The usual conventional complementary base pairing. What is complementary base pairing? Adenine pairs with uracil and cytosine pair with guanine occurs between the first two bases of codon and the last two bases of anticodon. Here, conventional complementary base pairing is very important in codon and anticodon recognition. The third base of the codon is rather flexible with regard to the complementary base. So the codon have complementary base pairing with the tRNA and mRNA. The third base of the codon is flexible. The anticodon region of tRNA consists of 7 nucleotides and it recognizes the 3-letter codon in mRNA. So this is the picture showing the codons and anticodon relationship. This is the codon sequence that is present in 5' to 3' and this is the anticodon sequence that is present in tRNA. The complementary base pairing is applied to first two base sequences which is present in the codon of mRNA and it is relaxed for the third codon which is present in the mRNA. Now we are going to discuss one important hypothesis, that is the Hubble's hypothesis. Hubble hypothesis put forth by Crick is the phenomena in which a single tRNA can recognize more than one codon. This is due to the fact that the third base in the codon often fails to recognize the specific complementary base in the anticodon, that is 5'. So this Hubble hypothesis was proposed by Crick. See, this is the picture explaining the Hubble hypothesis. This is the codon present in the mRNA from 5' to 3' and this is the anticodon present in tRNA. So tRNA and mRNA pairs in antiparallel direction, pairing at the first and the second codon and in accordance with the Wasson and Crick pairing rule, that is adenine pairs with uracil and guanine pairs with cytosine. But pairing rules are relaxed at the third place of the codon. Say for example, guanine if it is placed with cytosine or with uracil. So you can see the Hubble hypothesis in this. So Hubble hypothesis is that explains the degeneracy of the genetic code, that is existence of multiple codons for a single amino acids. Although there are 61 codons for amino acids, the number of tRNA is far less, that means around 40, which is due to Hubble. So there are only 40. Next, we move on to tRNA, the adapter molecule. The transfer RNA, tRNA molecule of a cell, acts as a vehicle that picks up the amino acid scattered through the cytoplasm and also reads specific codes of mRNA molecules. Hence, this tRNA is called as an adapter molecule. This term was postulated by Francis Crick. If you see the structure of tRNA, the tRNA has the clover leaf-like structure. It is synthesized in nucleus on a small part of DNA. In 1965, R.W. Hawley suggested the clover leaf model of tRNA. Though tRNA molecule consists of single strand, Each assumes a cloverleaf-like structure through folding. There are 3 folds in cloverleaf of tRNA. It has 4 arms, namely anticodon arm, D arm, TC arm, and amino acid acceptor arm. The tRNA molecule are made up of 73 to 93 ribonucleotides. The acceptor arm carries an amino acid. The anticodon arm has 3 anticodon nucleotide which will be joined with the anticodon of mRNA during protein synthesis. That is, 3 nucleotide in the tRNA pairs with 3 nucleotide of mRNA. In certain tRNA, in addition to these 4 arms, an extra arm called variable arm occurs as shown in this figure. The amino acid acceptor and anticodon arms are oriented in opposite direction. The process of addition of amino acid to tRNA is known as amino acylation or charging, and the resultant product is called as amino acyl tRNA or charged tRNA. Without amino acylation, tRNA is known as uncharged tRNA. That means if amino acid is added to tRNA, it is called charged tRNA. If amino acid is not attached to tRNA, it is called as uncharged tRNA. If 2 such tRNAs are brought together, polypeptide bond formation is favored energetically. Number of amino acids are joined by polypeptide bonds to form a polypeptide chain. It is nothing but a protein. This amino acylation is catalyzed by an enzyme called amino acyl tRNA synthetase. This is an endothermic reaction. That means it gives out a heat and it's associated with ATP hydrolysis. Breakdown of ATP takes place. 20 different amino acyl tRNA synthetases are known. The power to recognize codon on the mRNA lies in the tRNA and not in the attached amino acid molecule. The tRNA charged with amino acids serve as an adapter molecule to decode the information on mRNA. This is achieved by the interaction of tRNA with mRNA. The tRNA molecule has a region that contains complementary bases to the codon of the mRNA. For initiation, there is another specific tRNA that is referred to as initiator tRNA. There is no tRNAs for stop codons. The stop codons or the nonsense codons do not have the tRNA. Translation. Inside the body, the process of translation occurs with every single cell. Each cell has a nucleus. After transcription, mRNA move out of the nucleus and enter the cytoplasm. This mRNA strand acts as a template for protein synthesis. Present in the cytoplasm, an enzyme amino acyl tRNA synthetase. The enzyme macromolecule has 2 binding sites. One site recognizes the amino acid methionine. This is followed by the binding of ATP molecule and release pyrophosphate, resulting in activation of amino acid. Finally, the tRNA and the activated amino acid bind together. This amino acylated tRNA is known as met-tRNA and is released from the enzyme. Met-tRNA is released from the enzyme. This makes the commencement of the first stage of protein synthesis. This stage is called as the initiation stage. During the initiation stage, the small subunit of ribosome binds to the mRNA strand. The mRNA strand is made up of codons with 1 sequences of 3 bases. Then the ribosome subunit moves along the mRNA in 5' to 3' direction until they recognizes the AUG codon or the initiation codon. At this point, the met tRNA possessing the anticodon UAG pairs with to the AUG codon of the mRNA. Then, a large subunit of ribosome combines with small ribosomal subunit. The large subunit shows three sites: the acceptor site or A site, the peptidyl site or P site, and the exit site or E site. This whole unit forms the initiation complex. This is followed by the elongation stage. During this stage, another tRNA carrying a molecule of amino acid approaches the mRNA ribosomal complex and fits in the A site. Then a bond is formed between methionine and the amino acid molecule of the tRNA. As a result, the met tRNA becomes deacylated. The ribosome moves about one codon and the deacylated tRNA shifts to the E site from where it dissociates. Meanwhile, another tRNA carrying amino acid molecule attaches to the A site. This is followed by binding of a amino acid molecule. Repetition of this process leads to the formation of amino acid chain. So many tRNA are combined. This event is called elongation. Finally, the UAG codon or the stop codon reaches the A site and the elongation is terminated. Termination is the last stage of protein synthesis. A chain of amino acid molecule is released from the ribosome. This released amino acid chain is the protein, and this part of protein synthesis is known as translation. Then the tRNA detaches from the mRNA, the ribosome detaches and dissociates into small and large subunits. Regulation of gene expression has been extensively studied in bacterial cells which contains genomic DNA. These genes on transcription gives mRNA and by translation produce protein. Both processes are energy intensive. Only those genes involved in homeostasis are expressed continuously. They are called as constitutive expression. When this operon, most of the genes are controlled by expressive signals which are usually molecules in the growth medium. The signals that positively regulate the gene expression are called as activators, while the signals that negatively regulate gene expression are repressors. Activators and repressors are DNA binding protein that regulate the gene expression. The DNA sequence at which the repressor binds is called an operator, while the DNA sequence at which the activator binds is called as activator binding site. Now let's talk about lac operon. Operon is the functional unit of DNA containing cluster of genes under single promoter. This lac operon has three structural genes, namely LAC-Z, Y and LAC-A. The LAC-Z gene codes for beta-galactosidase, which breaks the lactose into glucose and galactose. LAC-Y genes codes for lactose permease, which transport lactose inside the cell. And LAC-A gene codes for thiogalactoside transacetylase, which removes the toxic substances transported by LAC-Y genes. And this LAC-I encodes a repressor that binds the operator and represses the lac operon. Let's see how this lac operon functions. In absence of lactose, the operator is occupied by the lac repressor. This ultimately shuts down the lac operon genes, basal level transcription of lac genes. Now, and sometimes even in the absence of lactose, the repressor may fall on from the operator, allowing the transfer of lac genes. This occurs because the binding of repressor with the operator is not permanent. This is always some amount of lactose permeases, beta-galactosidase and thiogalactoside transacetylase enzyme present in the cell. When lactose is present, lactose enters into the cell by lactose permease enzyme present in genes. Lactose binds with the repressor and it releases the repressor from the operator. So it releases the repressor from the operator. As the operator is free now, the RNA polymerase can now bind to the promoter and initiate the transcription of lac genes. Now, as lac genes are continuously transcribed, more amount of lactose permeases and beta-galactosidase are formed. More amount of lactose enters into the cell. So, this allows permeases and beta-galactosidase are formed. Lactose is hydrolyzed to form glucose and galactose by beta-galactosidase enzyme. Next we see what is Human Genome Project. Human Genome Project. By taking a part of a gene, we can able to find out or examine the genetic abnormalities of an individual. So the International Human Genome Project was launched in the year 1990. It was a mega project and took 13 years to complete. The human genome is about 25 times larger than the genome of any organism sequenced to date and is the first vertebrate genome to be completed. Because human is a vertebrate, so it is the first vertebrate genome to be completed. Human genome is said to have approximately 3 into 10 to the power of 9 base pairs. Human Genome Project was closely associated with the rapid development of new area in biology called the bioinformatics. What is bioinformatics? Getting information about the protein, nucleic acid, etc. in our body from the genes. So it is called as bioinformatics. This HGP is helpful for this bioinformatics. Next is the goals and the methodologies of Human Genome Project. So what are the goals? The main goals of this Human Genome Project are, it helps to identify all the genes. Already I told you there are different base pairs, 3 into 10 to the power of 9 base pairs. So it helps to identify all the genes in human DNA. It helps to determine the sequence of the 3 billion chemical base pairs that make up the human DNA. And it also helps to store this information in database. So these informations are stored in a database that is in computer. And this Human Genome Project improves the tools for data analysis. So it improves the tools, the means and the tools for data analysis, and transfer the related technologies to other sectors such as industries. So by getting the information, it helps to transfer this to the other tech sectors and address the ethical, legal and social issues that may arise from the project. So these are the goals of Human Genome Project. It's a very important 3-mark question. So the methodologies of the Human Genome Project involve 2 major approaches. One approach was focused on identifying all the genes that are expressed as RNA. They are called as expressed sequence tag, ETS. The first approach is ETS. The other approach was sequence annotation. Here sequencing the whole set of genome was taken that contains all the coding and the non-coding sequences, and later assigning the different regions in the sequences with functions. For sequencing, the total DNA from a cell is isolated and converted into random fragments of relatively smaller sizes and cloned in suitable host using specialized vectors. What are vectors? Vectors are used for transmitting these genes. So this cloning results in amplification of pieces of DNA fragments so that it could subsequently be sequenced with ease. Bacteria and yeast are the two commonly used hosts, and these vectors are called as. So we can use the bacteria and yeast as a vector. So they are called as BAC and YAC. That is bacterial artificial chromosome and yeast artificial chromosome. The fragments are sequenced using the DNA sequencer. The fragments are sequenced by using DNA sequencer, which was developed by Frederick Sanger. We can call it as a Sanger sequencer. The sequences are then arranged based on the few overlapping regions using unspecialized computer-based programs. So with help of this specialized computer-based program, they can sequence the overlapping regions. Overlapping means ஒன்று மேலே ஒன்று அடுக்கிறது. So these sequences are subsequently annotated or assigned to each chromosome. The genetic and the physical maps on the genome are assigned using the information on polymorphism of restriction endonuclease recognition sites and some repetitive DNA sequences called microsatellites. The latest method of sequencing even longer fragments is by a method called shotgun sequencing method. There are two methods. One is the microsatellite, another one is the shotgun sequencing method using supercomputers, which has replaced the traditional sequencing methods. Next, we move on to the salient features of Human Genome Project. Although Human Genome Project contains 3 billion nucleotide bases, the DNA sequences that encode protein makes up only 5% of the genome. An average gene consists of 3,000 bases, the largest known human gene being dystrophin with 2.4 million bases. And third one is the function of 50% of the genome is derived from transposable elements such as LINE, line, and ALU sequence, arithmetic logic unit sequence. Genes are distributed over 24 chromosomes. Chromosome 19 has the highest gene density. So which chromosome has highest gene density? Chromosome 19. Chromosome 13 and Y chromosome have lowest gene densities. The lowest gene density is chromosome 13 and the highest gene density is chromosome 19. The chromosomal organization of human genes shows diversity. That means difference in the characteristics. There may be 35,000 to 40,000 genes in the genome, and almost 99.9 nucleotide bases are exactly the same in all people. So most of this 99.9 nucleotide bases are same for all the people. Only 1% will change. The functions for over 50% of discovered gene are unknown. Less than 2% of the genome codes for protein. The repeated sequences make up very large portion of the human genome. The repeated sequences have no direct coding function, but they show the light on chromosome structure, dynamics, and evolution that it helps in genetic diversity. Chromosome 1 has 2,968 genes, whereas chromosome Y has 231 genes. There may be difference in the number of genes in chromosomes. Scientists have identified about 1.4 million locations where single base DNA differences, that means SNP, single nucleotide polymorphism, pronounced as we can call it as a SNP, SNIPS, occurs in humans. Identification of the SNPs is helpful in finding the chromosomal location for disease-associated sequences and tracing the human history. So these are the salient features of Human Genome Project, where you will get 1 marks and 2 marks, and it may be also asked in 5 marks. Next is the applications and the future challenges of this Human Genome Project. So what are the applications or what are the uses? The mapping of human chromosome is possible to examine a person's DNA and helps to identify, number 1, genetic abnormalities. Number 2, it is useful in diagnosing diseases. Number 3, it helps to provide genetic counseling to those planning to have children. And next one is, this kind of information also create possibilities for new gene therapy. You know about gene therapy. What is gene therapy? Transfer or changing of a defective gene by a normal gene. Besides providing clues to understand human biology, learning about non-human organisms, DNA sequences can lead to an understanding of their natural capabilities that can be applied towards solving challenges in healthcare, agriculture, energy production, and environmental remediation. So, what are the important advantages? What are the symptoms? Once the genetic sequence become easier to determine, some people may attempt to use this information to profit or for political power. And so many insurance companies may refuse to insure the people at genetic risk, and this would save the companies the expenses of future medical bills incurred by less than perfect people. Another fear is that attempts are being made to breed out certain genes of people from human population in order to create a perfect race. DNA fingerprinting. Each of us have the same chemical structure of DNA, but there are millions of differences in the DNA sequences of base pair. This makes the uniqueness among us so that each of us, except the identical twins, is different from each other genetically. The DNA of your person and the fingerprints are unique. There are 23 pairs of human chromosome with 1.5 million pairs of genes, and 3 billion base pairs are present in every DNA. And this, by comparing these DNA sequences, we can able to identify the DNA fingerprinting of the individuals. We can see the DNA by comparing this fingerprinting, we can find the DNA of this individual. In human, 99% of DNA base sequences are identical and are known as bulk genomic DNA, and remaining 1% of DNA base sequence differ and have repeated sequences known as repetitive DNA. 1% is repetitive and 99% is bulk genomic. And you can separate this bulk genomic and this repetitive DNA by a method called density gradient centrifugation. DNA fingerprinting involves in identifying differences in some specific regions of DNA sequence called repetitive DNA, because in this sequence a small stretch of DNA is repeated many times. This repetitive DNA are separated from bulk genomic DNA as different peaks. The bulk DNA forms a major peak and the other small peak is called as a satellite DNA. Depending on the composition, base composition, length of the segment and number of repetitive units, the satellite DNA is classified into two types, mini-satellite and microsatellite. These sequences do not code for any protein, but they form a large portion of human genome. These sequences show high degree of polymorphism and form the basis of DNA fingerprinting. DNA isolated from blood, hair, skin cells or other genetic evidences left at the scene of a crime can be compared through VNTR patterns with the DNA of the criminal. And this satellite DNA do not show any impact on producing ability and exhibit high degree of polymorphism originating in a population. DNA polymorphism is resulted either by mutation in somatic cells or by germ cells, which accumulates over generation. DNA polymorphism is the guiding principle behind the genetic mapping, therefore helps in DNA fingerprinting techniques. This DNA fingerprinting techniques was first developed by Alex Jeffer in 1985. This technique involves southern blot hybridization, which used radio-labeled VNTR as probe. VNTR is a small sequence of DNA arranged tandemly in several copies, which varies from one chromosome to another in an individual. A very high degree of polymorphism is seen in these repeats due to the size of VNTR, which varies from 0.1 to 20 kilobytes. Now, we will see the steps involved in DNA fingerprinting. First one is the extraction of DNA from a sample of blood or semen. Second one is the polymerization PCR, polymerase chain reaction. Many copies of DNA can be produced by PCR. Then fragmenting of DNA takes place. So it can be fragmented by restriction enzymes which cut the DNA into smaller fragments. Fourth step is the separation of DNA by electrophoresis. During electrophoresis, in an agarose gel, the DNA fragments are separated into bands of different sizes. The fifth one is the denaturing DNA by using alkaline chemicals or by heating. And sixth one is the blotting. The DNA band pattern in the gel is transferred to a thin nylon membrane placed over the size fractioned DNA strand by southern blotting. Seventh one, by using the probes to identify the DNA. So a radioactive probe is used to identify the DNA bands. And eighth one is the hybridization with that probe. After the probe hybridizes, the excess probe washed off, a photographic film is placed on the membrane, or X-ray film can be used on the membrane containing DNA hybrids. Ninth one is the exposure on film to make a genetic or a DNA fingerprint. The radioactive label exposes the film to form an image corresponding to the DNA bands. The thick and the thin dark bands forms a pattern of bars which constitute a genetic fingerprint. Today, the accuracy of DNA fingerprinting technique has further improved due to the advent of polymerase chain reaction PCR, while multiple copies of DNA sequence can be made by this PCR. DNA fingerprinting has also advanced due to the use of different types of DNA probes. DNA probes are used to form the different DNA fingerprinting. If you see the applications of DNA fingerprinting, it is used in the forensic labs. It can be used for the identification of a person involved in criminal activities. And it is also used for settling the paternity or maternity disputes and in determining the relationship for immigration purposes. And it is also used as a pedigree analysis for evolution, inherited patterns of genes through generation, and for detecting the inherited diseases. It is also used in conservation of wildlife, protection of endangered species of animals. And it is also used as an anthropological studies. It is useful in determining the origin and migration of human population and genetic diversities. So, these are the applications of DNA fingerprinting, which is used as forensic analysis, pedigree analysis, conservation of wildlife, and anthropological studies. Children, I hope you all can understand the concept of translation, human genome project, DNA fingerprinting. So, we come to an end of our fifth chapter, molecular genetics. This chapter is a little bit complicated, but it's very easy to read. Read the questions sent to you in the assignment. If you have any doubts, feel free to ask me. Don't waste your time. Everything will change. What we are facing now is not permanent. So, have faith in God. Be safe at home. Thank you.